In this podcast, we're going to look at skeletal muscle. Let me start the podcast by just going over this histological view of skeletal muscle, just because it makes some interesting points. The first thing I want to point out for you is something that we label as the A band and the I band. Now, in a typical histological section, the A band stains darker than the I band. You can see the A band staining darkly and the I band staining lighter. But you can also notice that the I band is bisected by a structure called the Z line. And we'll talk about this in a few minutes. The other thing I want to point out, skeletal muscle has peripherally located nuclei. And in this image, what we can see is a portion of one cell here, a portion of another cell here, a portion of a third cell near the bottom, and then a portion of a cell at the very bottom and a portion of another cell at the very top. The light lines you see in the middle is actually connective tissue separating the individual skeletal muscle cells. As we said, the A band stains dark in a typical histological preparation. The A band can rotate polarized light if you were looking at skeletal muscle with a polarization microscope. So the A band is anisotropic, and this is where the name A band comes from the anisotropic properties of the molecules in that band. And that's contrasted to the I band, which is isotropic. Isotropic means it's not birefringent. It does not rotate polarized light. So that's just a little bit of a history of where the A and I band come from for names. The M band is in the center of the A band. It's sometimes visible on a histological section. You can just maybe barely see it as this lighter area in the middle of the A band, this lighter area right in the middle of the dark band. The Z line, I think you can clearly see this darker thin band in the middle of the I band or the light band. Some special terms I'd like you to keep in mind as we talk about muscle. If we talk about a muscle fiber, we're talking about a muscle cell, and we give skeletal muscle cells the name myofiber, and a myofiber is much longer than it is wide. You may remember that from the first podcast. If we talk about a cardiac muscle cell, we give it the name myocyte. So we contrast myofiber from myocyte, a myofiber being a skeletal muscle cell, a myocyte being a cardiac muscle cell. Please don't confuse the term myofiber, which is a living cell, with the term collagen fiber. As you know, a collagen fiber is a large macromolecule, but it's not living. Also, don't confuse these with the terms myofibrils. Myofibrils are bundles of myofilaments, and the myofilaments are individual cytoskeletal filaments, either actin or myosin. We'll talk about that in a few minutes. If we talk about the muscle cell plasma membrane, we give it the name sarcolemma. Sarcolemma for muscle cell plasma membrane. And by the way, sarco is a term that can be used to refer to muscle. So if we say sarcoplasm, we're saying the cytoplasm of a muscle cell. If we say sarcoplasmic reticulum, we're talking about the ER, and particularly the smooth ER in muscle cells. If we say sarcosome, we're talking about mitochondria in muscle cells. So sarco means muscle. In striated muscle, we talk about a unit of contraction as being a sarcomere, and sarcomeres span from Z line to Z line, and we'll show you this in a minute or two. Individual lengths of sarcomeres can vary within skeletal muscle cells, and sarcomere length relates to the entire length of a muscle. There are different types of fibers in skeletal muscle cells. We talk about red muscle fibers and white muscle fibers, and we can define these by how they appear on a histological preparation. A red fiber has a rich vascular supply, whereas white fibers have a poorer vascular supply. White fibers are larger than red fibers. They're innervated by larger nerve cells as compared to smaller nerve fibers innervating red fibers. White fibers have a very strong contraction. It's fast and strong, but the fibers fatigue easily, as opposed to red fibers have a slower and weaker contraction, but they don't fatigue easily. You might think of migratory birds like geese. They have a lot of red muscle fibers in their flight muscles, for example. You can look at the rest of the table and see how you can define the sarcoplasmic reticulum, levels of mitochondria, myoglobin, energy, etc. in red and white fibers. I do want you to understand there's also an intermediate fiber that you can identify histologically sometimes that has characteristics between red and white fibers. And anatomically named muscles will have all three fiber types in constant and characteristic proportions. And for example, one anatomically named muscle will have a given concentration of red, white, and intermediate fibers, and that might differ from a different anatomically named muscle. And just to put this in perspective, in physiology, you'll talk about type 1 slow twitch fibers and type 2 fast twitch fibers. The type 1 or slow twitch fibers are the red fibers. The type 2 or fast twitch fibers are the white fibers. I want to say a few words about the connective tissue investments of skeletal muscle because these play an important role in transmitting the forces of contraction in muscle to, for example, the bones of a limb. And this is important because individual muscle cells don't usually extend for the entire length of a muscle. We can start by talking about the epimysium. And the epimysium is a dense, irregular connective tissue that surrounds an entire muscle. And so that's what's kind of depicted on the cartoon here. And if you think about what you did in gross anatomy as you were dissecting the muscles in the limb, if you were to take with your forceps and pick up a large individual muscle, anatomically named muscle, you'd be grabbing onto the epimysium. It's a dense, irregular connective tissue surrounding the entire muscle. From the epimysium, you get a perimysium, which is a less dense connective tissue. This is derived from the epimysium. It surrounds fascicles or bundles of muscle fibers. And remember, the fiber is a muscle cell. And from the perimysium, one has a endomysium, which is a looser connective tissue or reticular fibers. It's consisting basically of reticular fibers in the basal lamina. The endomysium surrounds an individual muscle cell. So if we were to go backwards on this diagram, here's an individual muscle cell surrounded by its endomysium. Here are a cluster of muscle cells surrounded by a perimysium, and then here are a bunch of these fascicles now surrounded by the epimysium. And so you can imagine that as this cell contracts, the forces of contraction focused through the connective tissue sheets all the way to the large muscle so that it can impinge on a bone and can cause the bone to move. It's because the connective tissue investments of the muscle cells, they're interconnected with each other and then with tendons, for example, that as the muscle flows into the bone. Let's talk a little bit about skeletal muscle in relation to structure and function. Before I do that, let me just remind you of something. You may have had this early in your medical school training in embryology, but remember that embryonic muscle cells, myoblasts. Embryonic myoblasts are cells with a single nucleus, and these myoblasts fuse to form myotubes. Because these myoblasts fuse into myotubes, these myotubes become the muscle cells, and the muscle cells will wind up being multinucleated. Satellite cells, on the other hand, are the regenerative cells for skeletal muscle. They maintain just a single nucleus. On this cartoon, we have labeled a sarcomere. I would like you to appreciate that the sarcomere extends from Z-line to Z-line. I use the term Z-line. You can say Z-line or Z-disc. Those are interchangeable. Remember, the Z-line bisects the I band. So you can say that a sarcomere contains one half of an I band, an entire A band, and then another half of an I band. So the sarcomere going from Z line to Z line shows that structure. Here is the A band, the dark band on a histological preparation. The H band is in the middle of the A band, and you can sometimes see in a histological section the H band is bisected by the M line, and the M line would be right here. It's not labeled on the cartoon, so this would be H band. Here is A band, H band, M band. So again, on this side, A band, H band, and M line. Here is the I band and the Z disc. And remember, sarcomere goes from Z line to Z line. Myofibrils are parallel to each other, and they extend the length of a muscle fiber or a muscle cell. And there are many myofibrils per cell. Their precise alignment is what gives the cross striations of light and dark bands in striated muscle. And a myofibril would equal many myofilaments of actin or myosin, or I should say actin and myosin. And the myofibrils are held in register by intermediate filaments. So, for example, molecules like desmin and vimentin are at the Z discs, and they help to hold the myofibrils in register, and molecules like dystrophin at the plasma membrane will help to hold the myofibrils in register. You may recall in the first podcast I had a pair of slides that I said went beyond the scope of our course, but they were meant to emphasize that many cytoskeletal proteins are important in maintaining the structural organization and hence function of striated muscle. Here is an electron micrograph, and I put this up, and I'm going to label each of the pieces parts if you want, because this is an important thing that you have to be able to do. I will almost guarantee you on shelf exams, and for sure on our exams, you're liable to see electron micrographs of muscle cells, and you'll be asked to name some of the structures that you see. So here we've got the sarcomere labeled going from Z line to Z line. Here we have the I band bisected by the Z line. Here we have an A band, and the A band is bisected by the M line. Now, you don't always see it on electron micrographs, but in the middle of the A band, you can sometimes get a sense of the H band. There are also structures that I'm pointing at here and here. These structures contribute to or make up what's called the T tubules or the transverse tubules. And we're going to look at those right now. The T tubules and the sarcoplasmic reticulum are essential for uniform contraction of a skeletal muscle cell. let me put in perspective this diagram. This diagram just shows a portion of a skeletal muscle cell. So here are bundles of myofibrils, like so, and each of the little stippled dots in the myofibrils would be the myofilaments. Let me highlight for you the labels. Here's the A band, like so, bisected by the H band. Here is part of a I band here. Here is another part of an I band here with the Z disc right in the middle. The Z disc right in the middle of the I bands. Now, what we're showing here is a portion of the plasma membrane of the muscle cell, and it is invaginating into a transverse tubule or a T-tubule. This invagination of the plasma membrane is extending deep into the cytoplasm of the skeletal muscle cell. And then highlighted on either side of the T-tubule are dilations here and here, or here and here, of the smooth ER. So the T-tubules or transverse tubules are invaginations of the plasma membrane. They're closely associated with terminal cisternae of the sarcoplasmic reticulum, and they form what's called a triad near the junction of the A and I band. It's called a triad because there is one component of the sarcoplasmic reticulum, a second component of the sarcoplasmic reticulum, and the T-tubule. So they form triads at the A-I junction. You may remember that the sarcoplasmic reticulum stores calcium when skeletal muscle cells are in a relaxed condition. You may also remember that as skeletal muscle cells contract, the plasma membrane depolarizes. There's a wave of depolarization on the plasma membrane that extends into the T-tubule system, into the transverse tubule, because that is an invagination of the plasma membrane. There are voltage-gated calcium release channels on the sarcoplasmic reticulum, such that when the T-tubule depolarizes, it causes the voltage-gated channels to open, and calcium in the sarcoplasmic reticulum will spill out into the cytoplasm, and that will cause the skeletal muscle cell to contract. Now, when the skeletal muscle cell relaxes, the calcium is actively transported back into the sarcoplasmic reticulum. So the T-tubules in the sarcoplasmic reticulum are essential for uniform contraction of skeletal muscle. Here on the high-power electron micrograph, you can see the T-tubule system, a portion of the transverse tubule, and on either side of it the dilated cisternae of the smooth endoplasmic reticulums. And you can also see some profiles of mitochondria and the contractile myofilaments on this high-power electron micrograph. Let's say a few words about the molecular organization of the myofibrils. Remember, the myofibrils are parallel arrays of thin and thick filaments. So the parallel arrays of the myofilaments. Myofilaments being an individual actin or myosin molecule. The thin filaments are actin. Actin thin filaments originate at the Z disc. They project to the center of adjacent sarcomeres. They're approximately 7 nanometers in diameter and perhaps a micron long. On the other hand, the thick filaments are composed of myosin two molecules. These interdigitate with the thin filaments. They are perhaps 15 nanometers in diameter and maybe 1.5 microns long. Now, I don't care so much if you remember the dimensions of these filaments, but the thin filament is actin, about half the diameter of the thick filament. I want you to know that the thick filament is myosin 2 and the thin filament is actin. This cartoon highlights some characteristics of the myofilaments. So let's look at the thin filament first on the top. So that would be highlighted like so, the thin filament. The thin filament is composed of F-actin molecules. There are two chains of F-actin molecules that wrap about each other, and they are wrapped around by tropomyosin and troponin. You may remember from your study of biochemistry that F-actin is filamentous actin, and what F-actin is essentially a polymerized G-actin molecules. So in this cartoon, each of the little brown balls, if you want, would be a molecule of G-actin. Actin has polarity because G-actin molecules, you may remember from biochemistry, are structurally asymmetric. There are two chains of tropomyosin that are wrapped around each other that wrap around the actin filaments. And the tropomyosin regulates the binding of actin and myosin. And you may recall that in addition to the tropomyosin and actin, there are troponin molecules that make up part of the thin filament. Troponin has several subunits. There's a troponin T that binds to tropomyosin. There's a troponin C that binds onto calcium, and there's a troponin I that inhibits the interactions between actin and myosin. The bottom part of the cartoon shows the thick filament. The thick filament consists of several hundred myosin molecules, and a myosin molecule consists of two identical heavy chains plus two pairs of light chains. Now the artist has drawn the myosin molecule, the two identical heavy chains, just as different shades of green, only to let you realize that they're two different heavy chains, but they're identical to each other. And then the light chains shown here. I don't care that you remember so much that there's light meromyosin and a heavy meromyosin. Those are defined by enzymatic cleavage of the molecules, and you may have looked at that in biochemistry, and you may recall there's an S1 and an S2 region of heavy meromyosin, and the S1 region of the molecule is the business end of the molecule. That region has ATPase activity, and it is that region that interacts with the thin filaments. I'd like to say something about the structural organization of myofilaments within the sarcomere. First thing I want to say is that structural organization is maintained by a variety of accessory proteins. So let's put this diagram in perspective. This is a sarcomere going from a Z line to a Z line. Here would be the A band, like so, with the myosin molecules, and here would be part of the I band with the actin molecules, and you can see just labeled some accessory proteins like alpha-actinin, CAP-Z, nebulin, titin, and we'll come back and talk about those in a minute or two. I want you to first understand that you have to be able to correlate diagrams that relate to a structure like this, to structures that you see on the bottom, to electron micrographs of sarcomere structure. These are questions that people like to ask on standardized exams. So, for example, you see regions like so, labeled as I band, H band, M band, A band. So you can imagine if you took a section right through here, the cross section, you'd see the little red dots. That would be the I band. On the other hand, if you took a section right here, you'd see a larger set of dots. That would represent the H band. On the other hand, if you took a section like this, where you're catching both the A band and the I band, you'd have a section like this where you'd have larger dots surrounded by smaller dots. And if you took a section through the M band, you'd have something that looks like this, the large dots with maybe a matrix surrounding them. Look for 1 second at this structure and then look at this micrograph. This is a cross section through the A band. Each thick filament is surrounded by 6 thin filaments. So each thick filament surrounded by 6 thin filaments. So a thick filament here, then 1, 2, 3, 4, 5, 6 thin filaments. So it's important that you be able to make that type of correlation if you see this on a standardized exam. Here is something about the organization of accessory proteins in a sarcomere. These serve to first stabilize the actin and also to organize myosin. Now remember, because actin has a polarity, there's a plus end to actin molecules and a minus end. The plus end of actin molecules binds into the Z disc, and it's bound to the Z disc by proteins like CAP-Z and alpha-actinin, and these proteins serve to stabilize the actin filament at the Z line and to prevent the actin filament from polymerizing any further. The minus end of the actin filaments is towards the M line, and it is capped by tropomodulin. And tropomodulin is there to prevent actin depolymerization. So these CAP-Z, alpha-actinin, and tropomodulin molecules are essentially functioning to stabilize actin. You might remember, as we talked about cytoskeleton, actin is a very dynamic molecule. It can polymerize and depolymerize fairly readily in the cytoplasm. So you must keep it stabilized to have structural organization to striated muscle. Actin filaments are also surrounded by molecules called nebulin, and that's what's shown here, these nebulin molecules surrounding the actin filaments. That helps to anchor them to the Z disc, and it also helps to hold them into parallel arrays at the Z disc. And then finally, there's a protein called titin, and the artist has drawn that specifically here to almost look like a spring. Titin is one of the largest proteins known. It's between 2.5 to 3 million Daltons in size, and it runs from the Z line all the way to the M line, and it plays a role, or it's thought to play a role, in the assembly of myosin thick filaments and in the elasticity and tension of sarcomeres. There are a variety of proteins associated with skeletal muscle cells. You need to know about these proteins from the point of view of integrating what you've learned in biochemistry with what you're going to learn in physiology, and you need to know about these because you're almost certainly going to see questions about these different proteins on licensing exams. We've talked about myosin. You need to know something about myomesin, titin, C protein, G-actin, CAP-Z, tropomodulin, tropomyosin, troponin, alpha-actinin, nebulin. So you need to, on your own, learn about these proteins and be sure you understand some of the basic characteristics of these proteins. I do want to point out for you troponin because there's some clinical significance here. There are different isozymes of troponin T and troponin I if you look at the troponins in skeletal muscle and in cardiac muscle. And if a patient presents in the ER having myocardial infarction, as you might expect, cardiac muscle cells are dying. Well, as the cardiac muscle cells die, they're spilling troponin. And so one of the biochemical assays that is done very quickly is to look at elevated troponin levels in the blood. If there are elevated troponin levels, if they're looking at the cardiac troponins, that would indicate a myocardial infarct. On the other hand, if there's an injury to skeletal muscle, there could be elevated troponin, and there you'd have to be looking at the troponin isozymes that are in skeletal muscle as opposed to cardiac muscle. So, for example, the troponins that are released in a myocardial infarct would be different than the troponins that might be released from a skeletal muscle injury. Skeletal muscle contracts as myofilaments slide past each other within a sarcomere, and this is something that Huxley presented in the middle of the 20th century, and it's been fairly well understood for a long time. Thin and thick filaments do not get shorter, rather the Z-disc move closer together as thin filaments slide past thick filaments. During contraction, there's a greater overlap between thick and thin filaments. The I band width will tend to be reduced. The H band will appear to disappear or be obliterated. The width of the A band will not change. And there is a temporal delay between contraction of adjacent sarcomeres, and that temporary delay is necessary to counteract equal and opposite forces in adjacent sarcomeres as contraction occurs. This cartoon shows a sarcomere, Z-line to Z-line, in a relaxed state on the top and in a contracted state at the bottom. Note the I band in the contracted state is shorter, the H band is obliterated or reduced, the width of the A band has not changed because you can see the width of the A band from the top to the bottom is not different. Clearly the width of the I band is much reduced, and the M band would still be present. The temporal delay or wave of contraction between adjacent sarcomeres occurs as individual motor units are recruited for contraction. So basically, as each sarcomere is contracting, there's a little bit of a delay going down the length of the muscle fiber. Contraction of skeletal muscle is all or none. Contraction is triggered by impulses from the myoneural junction, and the strength of contraction of a gross anatomical muscle is a function of the number of muscle fibers that are contracting. Contraction is regulated by motor units, and a motor unit is a single nerve cell plus all the muscle cells that it innervates. As you might imagine, muscles from fine motor control are innervated by individual nerve cells. So, for example, muscles that control the movement in the eye, one nerve cell might innervate one skeletal muscle cell. But on the other hand, muscles that are involved in the movement of gross anatomical components in the body, a single branching nerve fiber might innervate several hundred muscle cells. On the other hand, maybe the muscles that control movement of the fingers, one nerve cell may innervate maybe 10 or 20 individual muscle cells. As you might imagine, skeletal muscle receives both sensory and motor nerve input. This is a histological view of the myoneural junction for motor input. So what you're looking at histologically here would be a motor unit. So you've got individual skeletal muscle cells innervated by the branching nerve coming from this nerve that's innervating the muscle. Now, the motor fibers are myelinated axons of alpha motor neurons. You'll talk about that next year in neuroanatomy. But here is a higher power view of a motor end plate showing individual terminal boutons where the synapse occurs between the nerve cell and the muscle cell. And you'll talk about the physiology of contraction on these structures in your physiology class. Coordination of skeletal muscle movements depends on sensory nerve innervation. The central nervous system will monitor position of the limbs and the degree of muscle contraction. So we can talk about something like proprioception. Proprioception is an example. You close your eyes, you can always bring your hand up and touch your nose. Even if you're blindfolded, you can do that. That's proprioception, of knowing where the different parts of your body are in space. There are several structures that play a role in proprioception. One of these structures are called muscle spindles. Spindles are actually within the muscles themselves, and Golgi tendon organs, which are structures within the tendons. Muscle spindles monitor the length and length changes of muscle fibers as the cells contract, so it's measuring the muscle stretch or strain. Golgi tendon organs, which are in the tendon, monitor the intensity of contraction, and they can inhibit alpha motor neurons, so they can inhibit the nerve impulses coming into muscle cells. This is an example of a muscle spindle or a sense organ within a skeletal muscle, and the number of spindles varies in anatomically named muscles. The spindle is a modified or a collection of modified muscle cells that call intrafusal muscle fibers. These intrafusal muscle fibers, perhaps 8 to 10 of them, are surrounded by a space, a periaxial space, which is enclosed by a connective tissue capsule that's continuous with the perimysium and endomysium. On the diagram, you can also see the typical or ordinary extrafusal muscle fibers. There are several different varieties of intrafusal fibers. Physiology may go over the function of these. It's not that important for our course for you to know what these different intrafusal fibers are, but you may come across in your reading nuclear chain fibers, static nuclear bag fibers, and dynamic nuclear bag fibers. I only mention those in that you may come across them in your